Thanks, Henry. Good morning, everybody. Thank you for joining us. I'm Professor Gavins, and I'm the director for one of the research centres here at Brunel University, London. And this particular session is part of Brunel's Research Festival that's going on for the next month. There's lots of different things that you can dip in and out of. So have a look at the programme and do join us for other events. So the research centre that I direct is called the Centre for Information Research and Translational Medicine, or CIRTM as it's favourably known. Please feel free to check us out online, and by all means tweet if you'd like to. So the order or the agenda for today, I'm going to give you a little bit of an opening and introduction to the centre, tell you a little bit about the history behind it and the research that goes on within the centre. And then I'm going to open it up to different academics within CIRTM who are going to present a snapshot of the research that they're doing at present. So it gives you an idea of what's going on within the centre. Then after that, we're going to have a roundup, and any questions that you may have, feel free to pose them either in the chat or ask them directly and to any of our academics, and they'll do their best to answer them. Okay, so the centre, CIRTM, was established in September 2019, and it forms one of several research centres here at Brunel. The idea behind research centres really was to have a home in which academics could focus on their research and share very common themes within that particular centre, so that it could grow in critical mass and also sort of communicate and network with other scientists in their respective areas. So the specific objective for CIRTM was to bring together a critical mass of internationally outstanding basic and clinical researchers tackling today's global health challenges, and I'll talk about those in a moment. The mission is to promote not just interdisciplinary research, but multidisciplinary research, and you'll see this as we go through some of the talks, exactly how everybody is doing that here, both at sort of a local level within Brunel, nationally within the UK, and internationally, externally on the global platform that seems to be a little bit more accessible in today's world. So we do this research to provide an outstanding environment for the research training in the field. So we want continuity of what research is going on, but we also want to train those scientists of tomorrow. We wouldn't be able to do any of the work that we undertake within the centre without the generous funding from a number of different mechanisms. And so it's a big thank to those. We've named some of them here that you can see. And so we're very grateful to them for supporting the research that goes on within the centre. So I've said that we tackle global health problems. So those are health problems that not just within the UK, but affect billions of people worldwide. And we split them into mainly 6 areas, I would say, within the centre. We focus on cardiovascular diseases, inflammation that underpins a large body of diseases, infections and immunity, cancer, particularly rare forms of cancer, microbial resistance, and also our microbiome and health. So what happens in our gut and how that influences both our healthy environment as well as certain disease states. And you'll hear as we go through this morning, you'll hear some talks from researchers that are really performing cutting edge research within these particular areas. How do we tackle the challenges? We have a number of different ways we do this. We've just listed there some of the types of species that we use in order to be able to look at sort of physiological and pathophysiological mechanisms. So in other words, in organisms, it could be bacteria, it could be worms, zebrafish, mice, so we use rodents, and humans as well. So in some cases, we perhaps might take samples, clinical samples, or even do certain trials and things like that. So we really have a wide range of different approaches to tackle the challenges. And always we're thinking about, particularly when we use animals in research, we're thinking about the replacement, the refinement and reduction, or the 3 Rs as they're known, of animals in research, in order to make sure that we're using the lowest species possible to answer the scientific questions that we need to address. So we work across all biological dimensions to really understand, to provide an integrated understanding of both health and disease, the aspect of healthy aging. You know, we're all interested in being able to age in a healthy way so that it minimises potential risks for different disease states. We do this multi-layered approaches. We take from a genetic approach to molecular, cellular, organ networks, as well as population networks. And you'll hear more about this as you listen to the presentations this morning. I've said we do interdisciplinary research. We also do multidisciplinary research, different levels, different layers to answer and address those scientific problems that we're interested in. We take, for example, we integrate with different departments across Brunel as well as outside of Brunel. We look at, for example, big data analysis. We'll hear some talks on that. We look at use artificial intelligence in order to address some of that data. We've got some engineering. We'll hear about organ on a chip from Ashley a little bit later. Social sciences, we incorporate medical sciences, all of which enables us to really target and understand different disease states in a very specific manner. So that's it in a nutshell as far as the centre goes. That's what we do. But I think the best way to really appreciate the type of work that's going on is to listen to the experts within the centre. As I've said before, by all means, please do check us out online or tweet if you want to. If you've got any specific questions, feel free to also email me. Or any of the academics as we go through the talks if you're specifically interested in different areas. So without further ado, we're now going to listen to sort of 5-minute presentations from some of the academics within the centre, a snapshot of the type of research that we're performing at CIRTM, which we hope will whet the appetite. So feel free to ask any questions. So I'll kick off just because it makes it easier. I'm already standing here. So my lab, Gavin's lab, again, please feel free to check it out on the web, so gavins_lab.com. Specifically, my lab looks at inflammation and its resolution. So we're interested in what happens in health and disease, so again about aging, etc., but really from the microcirculatory point of view. So we're interested in what happens in our blood vessels, specifically our immune cells and what they do. So, you know, cells such as our white blood cells, such as neutrophils, and you can see here a nice example of a neutrophil in that vessel. So we tackle one of the most important drivers of disease, and that is inflammation. And we do that, and I'm sure that many of you are aware of different inflammation. I'm sure that everybody at some point will have had inflammation. And that's not necessarily a bad thing. It's the way in which the body is able to tackle pathogens or foreign substances that target it. So, for example, there's some examples here. You can see sort of a growing toenail. Perhaps it might be a sty on the eye, or you've got a spot and it fills with what we call pus, which is full of neutrophils. It's all doing a good positive thing. It's the body's way to tackle problems within it. And under normal circumstances, that is absolutely fine, and the body's able to restore homeostasis. But we're interested in what happens when that process is dysregulated, and that happens in certain diseases. And so we're interested in unraveling the complexities of immune responses in vascular inflammation to improve the treatment of human diseases. So as I say, under normal circumstances, you get this onset phase, it's inflammation, the body does, and then it resolves. Great, no problem. The issue is this red line that you can see when you get this excessive response that's non-resolving. And so what we do in my lab is look at this effect in different disease states and see if we can manipulate that profile, so that sort of blue dotted line, manipulate the profile with drug treatments, with pharmacological applications. Number of different diseases that have chronic inflammation, I've listed quite a few there. The ones in red are really the ones that we focus on in the lab, some in a lot more detail than others. So, for example, inflammatory bowel disease, ischemia reperfusion injury, which also incorporates transplantation, stroke, sickle cell disease, kidney disease, cardiovascular disease. So quite a number really of diseases, but they all have inflammation underpinning them. So we do this in order to, we look at the pathophysiology, the mechanisms, the pathways in those diseases in order to find precision medicine. So find better medicines that are more specific to you or to I, you know, when to help our bodies resolve from that inflammation. And this is classed as reperfusion pharmacology. So drugs that enabled us to target that pathway to resolve inflammation. Do it in multiple ways. We take data, we integrate that data. In some cases, we do what's called drug repurposing or repositioning. So drugs that are available on the market for one discipline may be applicable for other disease states. And we also find, or are interested in finding, novel targets that could help the body resolve inflammation. As I say, we're interested in neutrophils, this particular immune cell, this what we call a firefighter or a decision shaper that enables that sort of interaction between different cells, and it's heavily involved in inflammation and heavily involved, it's the switch for resolution. And so we're interested in that particular cell type, and we've looked at different mechanisms that can drive the resolution process by changing the functionality or the versatility of that cell to change the phenotype to drive inflammation resolution, particularly in cardiovascular disease. And we've just put in for a phase 2A trial on this particular area. Lots of different techniques and methods. Don't really need to know all of those. It's more for perhaps specific interest, I would say. But in particular, we take a lot of blood samples and isolate different cell types and look at the interactions with those cells. We use different species to do this: zebrafish, mice, rats, pigs, for example, and lots of experimental models of diseases. Some of the diseases, as I mentioned before. And we couple this with in vivo imaging, a lot of different techniques, and we also develop tracers to help us image image inflammation in disease. And finally, these are the funders that have supported us over the years or currently supporting us, to which we're very grateful. Okay, so that's my talk about my lab. I'm going to hand you over to Dr. Smith, who's going to talk about the work on tuberculosis. Steve.
Great, thanks very much, Felicity. So I am going to spend the next 5 minutes just very briefly giving an overview of the work that I've spent most of my sort of career in science been involved with. So as the title suggests, I'm an immunologist, and that means that I spend a lot of time looking at immune responses in the context of TB and vaccine protection against TB. So I'll say a little bit more about TB in a minute. But as an immunologist, there's anyone who's got any familiarity with the subject will know that there's a vast array of cells and molecules that make up our immune system. And this means that in terms of targets to investigate, there's certainly numerous. So the picture on the right hand side of this slide is actually some cupcakes made by a master's student in a class that I taught a few years ago. I took the picture because I always thought it would be a very useful way of illustrating the sort of variety of cells and molecules. So some of these I am actually interested in because they're important in TB, for example, macrophages. I think I'm not exactly sure what some of these are. So we have a T cell receptor, MHC molecules, antibodies as well. So one of the things about the immune response against TB is very complicated. So nearly every cell or molecule that you can think of is probably involved in some way or another. I've not got enough time to really go into TB in detail. It's a very complex disease. Until very recently, due to COVID, it was the most deadly infectious disease worldwide, only overtaken by COVID in 2020. The reasons for that are multifold. One of them is because we don't have a fully effective vaccine. So we do have a vaccine against TB. It's called the Bcg vaccine. You may well be familiar with it. Many people had it when they were young, either as babies or as teenagers. And TB does protect to an extent. And this is useful for us because it means we can look at BCG-induced protection and learn a lot from it. And hopefully this will teach us what we need to know when it comes to designing better vaccines that the aim of which is to get better protection against TB, especially in places where BCG doesn't work so much. So one of the main problems with BCG is that there are certain locations around the world where it's less effective. So this first part of the slide is just to show the different ways, generally speaking, that the immune system can protect us. This is sort of supposed to show what we call barrier function, and it means that a person who's exposed to the TB bacteria will not become infected at all. And so we know that there are certain characteristics, certain immune functions that can enable the body to resist infections, specifically speaking, the innate immune system, which works very rapidly and can eradicate TB before it's managed to get a hold in our lungs. However, many people do become infected. If they do, the best possible outcome is that they manage to contain the bacteria in the lung in what's known as a granuloma, a very complex structure of immune cells. And so a second, if you like, aim of a vaccine would be to make the body most efficient and best able to induce this protective granuloma structure. So these are the two possible, directions that a vaccine could go: inducing innate barrier function and very early immune responses, or inducing adaptive immunity that can protect us later in the infection and provide ongoing protection. If this doesn't happen, then the bacteria takes over and we get active tuberculosis. Okay, so just to say for the second part of my 5 minutes, exactly what I do in my group to try and investigate these problems. So this is a lot of the work that I've done was in my previous post at the London School of Hygiene. I was lucky enough to work in the group of Professor Hazel Dockrell for many years, and in that group we were very focused on the human immune response, and we used settings where we could obtain blood samples from individuals who received the Bcg vaccine and look for potential immune protective mechanisms in the blood of those individuals. So sometimes that was in adolescents who received BCG, a lot of the times it was in babies. So especially in the UK, BCG is mostly given to infants immediately after they are born. And this enables us to obtain blood samples, which are obviously very precious for very small babies. We can look at a lot of things in those blood samples, even though they're very small. Okay, so that leads us on to some techniques and methods using the group. So as you might expect, these are mostly to do with cells, molecules, proteins that you would be able to detect and find in a blood sample. So luckily, this does include a lot of the immune cells that we're interested in terms of a response against TB. So T and B lymphocytes, monocytes, and macrophages, for example. We can use techniques such as flow cytometry to investigate these cells in detail, what they look like on the surface, what molecules they express, and what proteins they secrete as well. We can also look at this secretion and measure proteins, molecules in the supernatant. So we use techniques like ELISA and ELISpot and Luminex to measure vast arrays of cytokines and chemokines and look into sort of chemokine profiles. So a lot of the power of these investigations comes when you look at many responses at once and look at marker profiles that can indicate either protection or susceptibility to disease. And then we also do some work with the bacteria itself. We can grow BCG. We're just developing a project with a collaborator who's got a BCG a version of the vaccine that's been modified to hopefully provide better protection by the way it grows once it's injected. And we can do functional assays as well. So one of the problems with this kind of work is it's very difficult to know when a response is actually going to protect against TB because we can't challenge a person with TB experimentally. So what you can do is develop in vitro assays where you can look at immune responses against model mycobacteria that step in for TB. So that's the way you look at that, is that the inhibition of growth that indicates when a response might be protected. And then lastly, I've also got an interest in the way some of the immune cells are programmed to respond better. So this is referring to work I did with a PhD student a few years back where he was interested in programming, epigenetic programming of innate immune cells and how they might sort of take on this capacity to protect better moving sort of forward following a vaccination. Most of the work that I've done when I was in the Dockrell group was funded by the European Union. I also managed to obtain my own funding, especially to look at some of these mechanisms that I mentioned in this slide. So, for example, from the Medical Research Council, specifically a network funded by the Medical Research Council called Validate, which I'm interested in vaccines against neglected diseases. And I've also had a bit of money from the MRC to pursue a collaboration with some a group in Korea a few years ago. Okay, that's all I have to say. Let's move on to the next presentation.
So hello everyone, and I'm I think the newest member of the centre, so it's great to be here. And like Steve, I was actually also working at the London School of Hygiene and Tropical Medicine until a couple of months ago. So I'm going to talk to you, I'm also an immunologist, and I'm particularly interested in the host immune response, and particularly about host variability in infectious diseases and how come some people are more susceptible to the same pathogen as other people are resistant. My work focuses on myalgic encephalomyelitis, chronic fatigue syndrome, which I'll go into a little bit more in the next slide, and also on tuberculosis. So about ME/CFS, which I'm going to say because it's a long name for myalgic encephalomyelitis, chronic fatigue syndrome. Essentially, most people when they suffer an infectious disease, they recover quite quickly, but for a certain proportion of people, they experience long-term symptoms, particularly long-term fatigue and post-exertional symptom exacerbation. So this isn't just being a bit tired, this is extreme tired, extreme exhaustion, and it can go on for months and years, and it's completely debilitating for some people. But we don't really understand why that is. So we've got here kind of a mixed population of people. Only some people will develop long-term post-viral fatigue, and this has particularly come to light and is more prominent in COVID era. So you will have heard of long COVID, and we don't really know why it is that some people recover quickly from a bout of COVID and some people go on and get long COVID, but we believe that for some people who have long COVID, there's actually similar mechanisms occurring as in ME/CFS. And this has importance for two reasons for my group. So we're interested as immunologists to understand what the mechanisms are, why some people resolve their infection and their inflammation and everything is fine and some people don't. But we're also interested in developing biomarkers. So what can we actually do with that knowledge and how will it help us? So as I say, the symptoms that were particularly associated with ME/CFS are chronic fatigue, post-exertional symptom exacerbation. That's not just tiredness, that includes brain fog, susceptibility to infections, so systemic disease, and we think there's probably a metabolic function going on, a dysfunction going on here long term. We have got some evidence of immunological abnormalities, so there's alterations in some of the T cell compartments post infection. And what our group is particularly interested in at the moment is the idea of human herpes viruses reactivating. So the human herpes viruses, there's 8 members of this family, and they're really quite prevalent. So in the adult population worldwide, a lot of us are chronically infected with these viruses, but most of us maintain them in a latent state, and we can control it with our immune system. And we've started getting some evidence that actually people with ME/CFS, their ability to control the herpes virus is reduced, and therefore there's more reactivation. And this is an area which we're particularly interested in following up. So the work that we do involves a lot of clinical sampling, and I've been very lucky to be working with this group, the CURE-ME group at LSHTM. And so these are the clinical partners that we work with, and they work with patient organisations to recruit participants into a biobank, and they go and talk to them, and they have a lot of symptom questionnaires, so we can actually follow what's going on with these people, and they take blood samples from them and biobank them, so we are able to work with them to access cells, so peripheral blood mononuclear cells, as well as serum, plasma and RNA samples. Work we've been doing recently looking at human herpes virus reactivation involves this sensitive method, droplet digital PCR, which is a way of really accurately quantifying the viral load that we have in saliva samples or blood samples. We also do quite a lot of flow cytometry, and I put this here as imaging is something that we want to be getting into that we haven't really quite got into yet, but these are the main tools that we're using. And these are the people who have funded us today on our ME/CFS work. Also, I'm interested in tuberculosis. I've been working in the TB field for a lot of years, over 20 years. And again, it comes down to the same kind of questions. Why do some people succumb to active TB, whereas most people don't? And Steve's really nicely highlighted that most people that are exposed to Mycobacterium tuberculosis either don't become infected or they maintain a latent infection. So only about 5 to 10% of people ever actually develop active disease. And the question is why? And what can we do about it? So I'm interested as an immunologist looking at the cells, particularly macrophages, and how they can control Mycobacterium tuberculosis. But we've also been doing a lot of work related to how we can detect whether someone is actually developing active TB and how they're responding to tuberculosis treatment. More recently, our work's been really focused on diabetes because people with diabetes have a threefold enhanced susceptibility to developing active TB once they're infected, but we don't really understand the mechanisms why that is occurring. So we've been doing some work with that, with a view about then what can we do about it. So the methods that we've used for our TB research are really quite similar. So we've been looking at clinical sampling. So we've taken blood samples from people with TB and TB diabetes. And I'm just highlighting here, I was involved in a consortium called Tandem, where we're actually collecting blood samples kind of worldwide from Peru, South Africa, Indonesia, and Europe. One of the things that I'm interested to do, now I've moved to Brunel, and anyway, with my research interest developing, is actually become a little bit more local. So there are people obviously in London who have TB and who have diabetes. So that's an area that I wanted to pursue further. Techniques that we've used, we've been doing a lot of gene expression work, initially with microarrays, and now we're doing more RNA-seq work, particularly our most recent work is TB diabetes, and also looking at the complement component C1q in macrophage function, how that might impact TB control. We do flow cytometry, we do ELISAs, we do a lot of real-time PCR. And put across through this. So these are ex-funders at the moment, and this is an area which we're not funded. So my work at the moment is predominantly with the MECFS, but I'm wanting to reinvigorate my TB research as well now I'm here. And that's it, thank you.
Okay, my research is about everything related to immunology, particular a molecule called EGR2, how this molecule regulate immune response. So early growth response gene, particularly we focused on EGR2 or 3, are the important molecule as we have shown is a lot of experience, a lot of evidence showing that in the regulation of immune response, so that the immune response, on one hand, efficiently function, but also control over react. So as you all know, my group is immunology, particularly interested in the adaptive immunity, which mainly focus on the immunology function is towards outside is control infection disease, and towards inside is prevent mutated cells, for example, cancer. Yeah. And particularly, we have been focused on 3 aspects. One is how to manipulate vaccine. And that is, we use a very specific, unique microsome vaccine. And that project was funded by MRC. And then we have been long-term interested in autoimmune disease. And mainly, oh, I don't know how to get, sorry. Mainly because we have our own autoimmune disease model. And another aspect which was very interested and very excited to us was we found this molecule is particularly important to control from the proliferation to the maturation or differentiation. So immune system is different from any other system. the immune system, the cells, they are normally not all are available. So it's kind of when that is needed, they start to proliferate and then differentiate. So it's not like a brain cells, we all have it, to heart cells, we all have it. So what we found, this molecule is really important from when it is proliferated, go into proliferation. So it's very interesting to know that the molecule, when it is knocked out, the cells, immune cells, will not be able to proliferate. Although some of the cells, they will be over differentiate. And the particular that is happened in the knockout. And then when we overexpress this molecule, then we found the cells, immune cells, can be over or very efficient to proliferate, but very difficult to differentiate. So unfunction, yeah. So they a lot of cells, but they don't do the work. So for that, we will found something that we believe this molecule is the link between proliferation and the differentiation. That was made MRC founded for this, the third part of the project. So the strategy we have been used mainly are transgenic lines. We have both EGR2/3 knockout. We have EGR2/3 overexpressed mouse line, and we also have EGR2 knock-in mouse line. And recently, we also developed a double T-beta and EGR2 knock-in and T-beta knock-in EGR2. EGR2 knockout. So with those mouse lines, we use the in vivo experiment system, like autoimmune disease, which it's a mouse knockout, EGR2/3 knockout itself is autoimmune disease mouse model. And then we also quite often use a virus infection model using Vaccinia virus. The other is we also established using B16, MC38, melanoma, and colon cancer cell, tumor cells to test how the immune response in this condition. So with my very close collaboration in Quimari, we have a lot of in vitro study with the fact sorting or facts analyze, and recently we are mostly focused on the RNA sequencing and chap-chap sequencing to profile the transcription profile. So recently, we are more focused on two projects. One is we try to see in the cancer condition how these two molecular, EGR2 molecular, play the role. We already know how it is a function in the virus infection condition. So we want to verify if a similar situation also could happen in the tumor. The other is more we are will be focused in the future is to see how EGR2 and T-beta regulate immune response. And T-beta for sure, that is an important molecule for the adaptive immunity. So I have been here 20 years, more than 20 years, more or less. And so 13 PhD students from Brunel have been graduated from those research projects. And we are interested to send more grant application. Mostly what we have planned is related to the autoimmune disease. If the timing, if the exam marking, we can manage it, and we send on the July, otherwise October we are aiming for. So currently I don't have myself grants available for my research. So some of the project which mainly rely on my close collaborator, Quimadi research grant. So that is all from me, and that's.
Okay, so unfortunately, Professor Sala Arturo couldn't be here today as he has COVID, but he did kindly share his slides last week. And so whilst I probably won't do them justice, I thought I'd just quickly run through them so you get an idea of some of the research that Arturo is doing within the centre. So Arturo focuses on molecular and cellular engineering to develop therapeutic approaches specifically for rare diseases and cancer. So he focuses on cancer such as neuroblastoma, adenoid cystic carcinoma, and for genetic diseases, Friedreich's ataxia. His team uses a number of different techniques in order to address questions that still arise from why these, for example, why certain cancers develop in certain populations, the genetic makeup for those diseases, and whether we can understand the pathophysiology in order to provide potential therapeutic targets. So he uses an array of different techniques to do that, from transformation assays to luciferase assays, PCR, CRISPR gene editing. For his genetic diseases, the Friedreich's ataxia, he uses a number of different approaches, the antiviral infection, he has transplantation models, behavioral assays, and histopathology. And these are all different approaches in order to really fully investigate and understand diseases that affect a large number of the population globally. So he's very well funded. He's received a number of different funds from quite a wide range of different charities, and from the MRC all the way to Great Ormond Street Hospital charity, all of which really understand the importance of the work that Arturo and his team are doing, and are trying to, Arturo is trying to address these specific aspects. So he's very well funded, but unfortunately he himself has currently tested positive for COVID. Okay, passing on to Ashley.
Morning, everyone. So my research, I'm going to talk a little bit about one section, which is developing an organ on a chip. But a lot of my research, I'm interested in microbiology, I'm interested in microbial communities, in various situations, not just the organ on a chip and bacterial vaginosis, but I have links with other projects where we're looking at the gut microbiome, looking at links with cancer, we're looking at plastic degradation, which Ronan McCarthy might talk about in a minute. But what I'm focusing on, one main area of my research, which I'm driving forward, is when bacterial vaginosis is the most common infection of the vagina for women. And it's a neglected subject, and there's a lot not known about bacterial vaginosis. It was historically thought to be caused by Gardnerella, but with the advent of next generation sequencing, so looking at microbial communities and understanding the bacteria that colonize the vagina, that's been put into question because Gardnerella is also present in healthy women. So the one problem with bacterial vaginosis is there are no real models that can be used to study bacterial vaginosis. We can't really experiment on humans to look at what can trigger bacterial vaginosis or what can cause Gardnerella, which is the most common pathogen associated, to overgrow, which causes bacterial vaginosis. But we try to find models. There are mouse models, but they're not particularly good because one of the bacteria that colonize the vagina predominantly in healthy women or women without bacterial vaginosis, which you really should say, is lactobacillus. The mouse vagina doesn't really have lactobacillus in it, so we can't use it as a proper model. So what we're doing, what I'm doing is I'm trying to make an organ on a chip. This sounds a bit Frankenstein science, but what we're actually doing is we're trying to grow human cells. A lot of tissue culture work using human cells in single layer. What we're trying to do is get them to grow in 3 dimensions so that they properly represent the communication that goes on with cells, because a single layer they don't communicate properly, and then they can potentially start differentiating when you have them in 3 dimensions. There are many approaches that have been attempted to grow tissue culture cells in 3 dimensions. NASA have done stuff where they've had a sort of circular incubator where they sort of get them suspended. What we are doing is we are getting basically a tiny chamber and we're putting a membrane in. And it's not like a flat membrane. It's, I suppose, a bit like cotton wool. It's a sort of candy floss, which has got lots of holes in it. The cells can grow in amongst and around these. And I have a PhD student, Shuai, who's working on this, and he is trying to spin these membranes. So he's got this technical candy floss machine, I call it, where he's creating different membranes using different compounds, which will biodegrade over time, because we want them to basically go. We don't want them hanging around. We don't want plastic waste or problems like this. And we're trying to find a suitable membrane that we can use to do this. Once we've got this working, once he's done this, we can then combine it into this chip, which is like a tiny chamber, and we'll grow the cells. And I've had a placement student who's been working a lot on characterizing different bacterial species that are associated with the vagina and getting and optimizing their growth into tissue culture conditions. Because bacteria have their own optimal conditions, but tissue culture is completely different. And if I mention bacteria and tissue culture, people hate me, but I keep well away, so don't worry on that. And also, with part of this, pathogens in clinical settings, there's lots of pathogens people detect by growing them, different things like that. I've been doing some work with another PhD student and my placement student on trying to use protocols to do rapid detection of pathogens. This is, I don't know if you've heard of LAMP PCR. This is a very quick PCR, takes 30 minutes, doesn't involve anything fancy. It just involves an incubator at 65 degrees. And we've been looking at a rapid protocol to try and detect pathogens. Aaron's been sort of more associated with trying to do work on COVID and other pathogens in the poultry industry. And my placement student has been doing the same. And we're trying to find a quick way that we potentially take a swab from someone and just in a clinical setting, in a doctor's surgery, or in the developing world, we can quickly do a swab and identify whether these pathogens are present in half an hour without any real technical equipment, rather than having to rely on high-tech approaches like PCR and things like that. So the key techniques, I haven't gone into as much detail as everyone else. Molecular microbiology, everything associated with microbiology in the lab. I've done most techniques over the times. I've got a lot of experience with next generation sequencing. So that's from the lab taking samples all the way through to the bioinformatic output. So I do both the wet science and the bioinformatics on this. I've also got experience of doing tissue culture infection studies, which is going to be partially sort of introducing the bacteria with tissue culture, see whether they internalize into cells or whether they just colonize the surface. Obviously, I've talked about Shuai, my student, with electric spinning membranes. These are creating artificial membranes with designated pore size, density, and things like that. We're also doing 3D printing. This is obviously to make the chips that all this organ on a chip is going together. So we've got experience, Ruth Mackay, who's over in engineering, who have strong links, and Elizabeth Silva are the other 2 PIs who are associated with this. They're not in CIRTM. And the funding, not a huge amount yet. I've had my brief award. I'm part of an EU Horizon grant with Ronan on plastic degradation. My PhD student Shuai is funded by the EPSRC. No, that's Aaron, sorry. And Shuai has got a scholarship from China, Zhuang University of Technology for this. So yeah, thank you very much. I'll hand over to Ronan.
So thank you very much, Ashley, for passing over. So my research focuses in on bacterial infection and the resistance of bacteria to antibiotic treatments. And I think it's something that generationally, is quite overlooked, because if you go back to the 1900s, we had a situation where in almost every single age demographic, the leading cause of death was infection. We then obviously had the advent of antibiotics, and that completely changed everything. And things that could routinely kill people were now very easily treatable. However, over, I suppose, since the 1970s, 80s, and more into the 90s, we've started to run out of antibiotics, and the antibiotic discovery pipeline has essentially dried up. And this has created a very problematic situation because, as we're all well aware, bacteria are incredibly adept at evolving. They can evolve to almost every single niche on this planet, and equally they can evolve resistance to different antibiotics incredibly quickly. And on average, it's about between 3 to 5 years, if you're lucky, between the introduction of an antibiotic to the clinic to the evidence of resistance within the clinic. In some instances, it's much, much quicker than that. That's creating a massive, massive problem in terms of treatment. And when you consider the different situations whereby a person is given an antibiotic, most forms of surgery, people are given a prophylactic antibiotic to try and minimize the risk of infection, even down to simple things like a tooth cavity. You're given antibiotics to potentially prevent an abscess. Now, the problem with that is more and more, and many of you may have experienced it yourselves, you're encountering a situation where the first antibiotic you're prescribed doesn't work. What you have to do is go back to the doctor, and the doctor will give you a second antibiotic. And that cycle can continue, and it can continue, particularly in a hospital environment if somebody's got a hospital-associated infection, such that there is no antibiotics that will work for the particular infection that you have. And that's happening more and more in hospitals, where you get pan-drug resistant infections, where the infection that is taking place is resistant to every drug available to humans. So what my research focuses in on is a couple of different avenues. It's looking at, firstly, how do bacteria evolve resistance, and can we try and stop that? And secondly, then, can we identify new compounds or new molecules that will potentially stop bacteria or bacterial infections? So we focus in on a couple of different techniques. In terms of the pathogens we work on a wide variety of different ones, but I would say we specialize largely on Pseudomonas aeruginosa and a particular pathogen called Acinetobacter baumannii. Now, the reason we focus in on them is partly because they're number 1 and 2 in the World Health Organization's list of priority pathogens that are in urgent need of novel therapeutic developments. So the main strategies that we do is largely high throughput drug discovery. So we have a number of different robotic platforms that we use for this, and we have a range of different collaborators. And we do this on 2 fronts. One is looking in diverse chemical libraries for potential novel compounds that will either inhibit the growth of bacteria, or we also look for something called a next generation antibiotic. And that's something that actually doesn't kill the bacteria, but essentially disables the mechanisms that it uses to establish infection. Now, the idea with these next generation antibiotics is that it's very hard then for a bacteria to evolve resistance to it, because the selection pressure is quite mild. We also then look at resistance determinants. So we look at the genes that are involved in allowing a bacteria to become resistant to infection. We look at the spread of these genes, and we look critically at how we can potentially disable these genes. So say, for example, bacteria have genes that encode efflux pumps. These efflux pumps are very critically linked to resistance. When they're upregulated, they can essentially pump the antibiotic out of the cell quicker than the antibiotic can be toxic. But you can block these pumps, just like any pump in the real world. You can block it with specific molecules, and if you disrupt those efflux pumps, oftentimes you can resensitize the bacteria. One area that we're very, very interested in also is biofilm formation, because there's a bit of a misconception around bacterial infections, in the sense that we often think bacteria are free floating within the body and they're very rarely settled in one area. And this is very far from the truth, because what often happens in about 80% of hospital-associated infections is bacteria will form a biofilm. What this biofilm is a community, because just like humans or animals, when you work together, you've better resources and you're better protected from the environment, and equally you can share resources among each other. That's exactly what bacteria do when they form a biofilm. So within that biofilm, you have a wide range of genetic heterogeneity, but also metabolic heterogeneity, whereby different bacteria are shutting down their metabolic systems to overcome antibiotic treatment. So we study how bacteria do this and if we can stop it, because again, if they're forming a biofilm, they're between 10 to 1,000 times more resistant to antibiotic therapy than they are if they were singularly floating. The other thing we do quite a bit is look at model organisms. So we really drive this insect model of infection called the Galeria melanella model. It can be used for a whole range of different types of infection, ranging from TB to Acinetobacter to Pseudomonas. And we've also recently developed, for the first time, a wound model of infection using these Galeria model. And we worked with a range of burn wound clinicians to show that this model can recapitulate what you see in human burns or mouse burns. We also look at the protective role of the microbiome. So we look at the commensal species that have a pathogen exclusion effect within our body, and we try to see, can we, I suppose, harness their protective power to prevent infection. The idea behind that then is potentially you can limit the use of antibiotics by potentially using protective species. So that, let's say, if you have a wound, you could try and populate that wound with commensal bacteria that will provide a pathogen exclusion effect. So in terms of funding, we've some work that's supported by the BBSRC, the Academy of Medical Sciences. The Galeria work is largely supported by the NC3Rs. We also have some work on Gardnerella Vaginalis, which was funded by Innovate UK. And we also have a plastic degradation project where we try and utilize again, this bacterial ability to evolve in any specific niche to see, can we get bacteria to evolve to actually eat plastic, hopefully overcoming quite a global waste problem. So that's just a summary of some of the work that we have done in the lab. So I will pass forward to Ansa. Thank you.
My research is focused around the host pathogen interaction in tuberculosis. As you know that someone has already, my colleague has explained about the tuberculosis. Just to go it again, the tuberculosis is an infectious disease which affects in 80% cases, the target is the lungs. In 20% of the cases, it is extrapulmonary. So once the infection goes inside the body, there is a 10%, 5 to 10% chances are there the body will develop the active tuberculosis, whereas in 90% of the cases, it remains latent. But if the immune response goes down because of the, say, for example, immunosuppressive treatment, then the reactivation of TB is there. 1/3 world population is infected, and therefore there is a huge reservoir for reactivation of TB. When the TB is co-infected with the HIV, there is a high mortality rate because the HIV targets CD4 and CD4 are the main cells which protect against the TB. Apart from that, there is a huge rise of multi-drug resistant TB. This is one of the factors, is because the TB treatment is prolonged. The drug, you have to take the 4 drugs for 6 months. First 2 months, there are 4 drugs, then the remaining 4 months, you have to take 2 drugs. But the patients, when they take for 2 months, they become all right, they feel fine, they just give up the treatment, and therefore the they don't take the treatment, and therefore there is a resistance developed when they start taking the drug again. And the other thing is that now there is also the cases of the resistant tuberculosis, which can be transmitted to others. The other thing is the protection is mainly by the cellular immunity, which is mainly CD4 and CD8 and other cells. Okay, unlike other infection, viral and other bacterial infection, where the antibodies plays an important role. Therefore, if you are trying to make a vaccine, you have to develop the epitopes, which are more essential to activate the cellular immunity rather than the antibodies. Because although the antibody plays a role, but they do not hugely protect. Because if you transfer the immunized serum from the one animal to another live animal, okay, then if you challenge it, it doesn't protect, and therefore the serum is less important. Now we have got the BCG vaccination, okay, but this BCG vaccination provides some protection against the child form of TB, but when the person becomes adult, it doesn't provide any protection. Okay, so my research is mainly focused on developing the new diagnostic test. It also involved targeting, producing some drugs or therapeutic interventions against the tuberculosis infection. And my interest was also to develop some diagnostic tests as well as to develop some vaccine. So these are my interests. So as you can see that the active research which are taking place are I'm using some proteins. Now, the body produces a lot of proteins which are able to control the infection. They play some role in protecting the infection. And since these are produced by the body itself, there is no immunological reaction. So if you take the serum, there are hundreds of the proteins circulating over there. So we know a number of functions of these proteins, what they are doing. But my interest is the functions which are not known, particularly I'm interested in the protein functions which are against the microbial infection and other pathogens. So the focus is on that side. Although we know, for example, complement protein C1q, we know that it is involved in the classical activation of complement system. But I am interested, how does the C1q is interfering or inhibiting the growth of Mycobacterium. For that, we are using the Bcg and Mycobacterium smegmatis as a model pathogen. And similarly, I'm using a number of other molecules like SPA, SPD. SPD is produced within the lung itself. C1q also is produced by the macrophage within the lung. Apart from that, whenever there is inflammation, there's a leakage, and therefore the proteins comes out, and therefore it is highly likely they will interact with the pathogen, and therefore we want to see what this pathogen these protein and pathogen interaction mean in terms of the bacterial growth. So we have recently published what this PAF C16, which is a lipid, which is again produced at the site of inflammation. How does it inhibit the growth? We have shown that this chemical is directly inhibiting the mycobacterial growth, and that has been published. And we have dissected out what is the underlying mechanism. It is damaging the cell membrane, and therefore it is inhibiting the growth of the both BCG as well as MX-matis. We are now on our way to reproduce these results in the actual pathogen, which is Mycobacterium tuberculosis. Apart from that, we have also shown that this lyso-PAF and its isotype, its variant, okay, they are also able to inhibit it. Okay, so we will take this into the animal model to see whether they are preventing it. Okay, so when the phagocytic cells are infected, this lyso-PAF also is inhibiting the growth inside the macrophages, not only directly, but also inside the macrophages. Now, the second most interest is the granuloma formation. The granuloma is the focus of control of infection in the body. If we don't have formed a good granuloma, the TB is not being controlled. For example, in HIV, patients with target is CD4 T cell, granuloma is not being found, and therefore the infection is not controlled. Similarly, if you neutralize the antibody like TNF, you use in case of rheumatoid arthritis, you use the antibody against the TNF. So in order to prevent the rheumatoid arthritis manifestation, the reactivation of the TB occurs because the granuloma formation is disturbed. So we want to make a granuloma in vitro, okay, by taking the PBMCs and infecting them with the BCG, because we have the facility for it, and the Mycobacterium escumatis to form the granuloma. And people have developed the granuloma, but what we are interested is whether this granuloma has the capability to inhibit the growth. Just forming a collection of the cells is not the aim. The aim is to show the functionality of the granuloma, whether it can inhibit the growth, which nobody has shown yet. So the interest is the identifying the new antigen from Mycobacterium tuberculosis. There is a huge 10% of the genome is devoted to PP and PE and PPE, and the function of these genes are some of them have been shown, some of them has not been shown. We want to identify some of those which have been published or which are not known, to identify the particular gene, whether they can be used for the diagnostic purpose as well as for the vaccine purpose. Previously, we have shown that the ESET6 and CFP, which are highly immunogenic, and that we used to show that can be used for the diagnostic purpose. And on that basis, we have launched a T-spot test, which is diagnosing TB infection, which is available in the world. Apart from that, we have also we are identifying the TB antigen that can be used for the vaccination against tuberculosis. Previously, we have shown that the MVA-85 which we produced, and we did a lot of trial, but this vaccine ultimately, when it went to phase 2B trial, that failed. But we believe still that there are other antigens which are highly immunogenic, which will provide the protection. Okay. So we are working on identifying the new antigen that may help not only for the diagnostic purpose, but also that can be used for the vaccine purpose also. So I will end over here. Thank you very much.
Well, good morning, everyone. So I just wanted to give an overview of what's going on in my lab. I'm also interested in tuberculosis. I've been working on TB for many years. Also interested in host pathogen interactions, but I'm coming at it from a different angle versus my esteemed colleagues on TB at Brunel. I'm coming from the angle of strain diversity and what is happening with the pathogen and how that can inform us in terms of how TB pathogenesis occurs. So my main background has been molecular epidemiology. I trained at Oxford University where I identified some biomarkers for studying pneumocystis carinii pneumonia, one of the AIDS-defining illnesses. I used those kinds of expertise to apply to tuberculosis, where I did a series of postdocs in California at Stanford University.
And essentially what I have been looking at is characterizing strain diversity in tuberculosis, particularly at the strains around the world. And that has allowed us to identify very interesting strains that are associated with transmission, those that are prone to reactivation versus latent TB infection, and active disease, and also drug resistance. Some of the major issues that happen in tuberculosis. And once we've identified these strains, with the explosion in genome technology, we looked at the genomes more closely. And we used microarrays back in around 20 years ago, where we identified large-scale deletions within the genomes of these strains. And obviously genes were disrupted within the bacterial strains, and we wanted to understand why. So we identified markers as a result of this that we could use for long-scale evolutionary studies, which have allowed us to determine that there are certain types or strains of tuberculosis that are associated stably with human populations. So what we have in tuberculosis, because M. tuberculosis, unlike most bacteria, are very clonal. They have very low variation. And a lot of the variation that we have is not through point mutations or mobile genetic elements. It's through rearrangement of genes as a result of insertion sequences. And that causes disruption of the genome, but also allows for genetic diversity for the organism to evolve. So we have a situation of host pathogen co-evolution, where we have certain strains that are associated with East Asia, the European continent, Oceania, and other places in the world that seem to predominantly infect these human populations in those areas. Okay, and we want to understand why that is. So that's really my goal in the work that I do. So what I've been doing as part of that is looking at PPE proteins. And so I'll mention these are proteins which are around 7% of the MTB genome, and we've characterized a few of them over the years, but most of them we don't really understand what they do. And they are very they're very repetitive regions. There's a lot of diversity of these genes, so they're very hard to study. And we've looked at how these proteins behave within this stable host pathogen co-evolution. And we've identified some PPE proteins that seem to be only in particular strains that are associated with these populations around the world. So this could be a fundamental part of the molecular dialogue between host and pathogen that underpin tuberculosis. So we're looking at that more closely. I have a very talented PhD student at the moment, and we've taken one of these proteins and tried to express it in the lab. And that is very exciting because these proteins are very difficult to work with. And we are in the process of characterizing the function of one of these PPE proteins. So the other things that I've been interested in also include developing novel therapies against tuberculosis using nanotechnology, particularly trying to use innate immune molecules, boosting innate immunity against TB as an alternative to using drugs, therapies, and avoiding drug resistance using nanotechnology. So that's mainly my human TB interest, but I've also been doing some work on bovine tuberculosis as well. Bovine TB is a major problem in the UK and in some other parts of the world, and it results in huge economic costs in agriculture. So we need better ways to be able to manage this, including an effective vaccine, which we don't have. So one of the ways we wanted to approach this is to boost innate immunity. There is a collecting important innate immune molecule called conglutinin in cattle, which is unique to cattle. This is very much related to surfactant proteins. And the function of this innate immune molecule is not really understood. But we have done some groundbreaking work initially to characterize its interactions with mycobacteria and macrophages, and found that it's able to inhibit infection of M. bovis Bcg, which is related to M. bovis, which infects cattle and causes tuberculosis in cattle. So if we can find ways to boost conglutinin, perhaps using it as an aerosol prophylaxis in the lungs, we may be able to prevent bovine TB. I'm also interested in the dynamics of infection in bovine TB as well, and looking for more prognostic markers, but that's in a sort of early phase of my work. So that's in a nutshell what I'm interested in. Thank you.
Yeah, do you need me to do the slides?
Yes, please, if you could move the slides, I would appreciate it. So yeah, good morning, everyone. My name is Hania Yagutka, and I joined Brunel just less than 8 months ago. I'm trained to be a medical doctor, but I wasn't interested in practicing medicine. I was more interested in doing research. So that's why I'm here. I'm interested in obesity, and so high BMI is a shared risk factor for all the disease that you are studying in this research center. And I'm interested to basically use genetics to understand what puts people at a higher risk of becoming overweight or obese, and what mechanisms link obesity to a list of these expanding the list of disease, for example, cardiovascular disease, diabetes, many types of cancer, also arthritis, and etc. So if you could move to the next slide, please. So I use different source of data sets. For example, I use metabolic biomarkers. I use anthropometric traits, for example, body fat percentage, body mass index, height, etc. And also a very valuable source of data that I use comes from MRI scans of UK Biobank participants. And through my collaboration with Professor Jimmy Bell at University of Westminster, and also 2 companies, I have access to a very detailed phenotypes measured from these MRI scans. I have data for from more than 50,000 individuals, and the target is to scan 100,000 individuals, and we are receiving more and more data. And basically, this data includes very specific measures of ectopic fat, for example, fat in the liver, pancreas, pericardial, for example, fat deposition. It also includes organ volume, for example, kidney size, pancreas size, spleen, etc., and also iron deposition in different organs. And I also use genetic data, and I combine all these data and use very specific genetic basically methods, and also machine learning methods to understand the genetic predisposition to risk of being obese or overweight, and also identify genetic background for diabetes, different types of diabetes, mainly type 2 diabetes, and cardiovascular disease. And the other aim of my research to identify causal risk factors for these disease outcomes. But more specifically, and recently, I've been spending time identifying subtypes of obesity. And specifically, my research recently has shown that obesity is a heterogeneous phenotype, and we identified a subtype of obesity, which I call it favorable obesity or adiposity. And basically, we have shown that people who carry a certain genes, they are able to store their extra fat in a safe place, which is subcutaneous adipose tissue, and therefore they are at lower risk of developing metabolic disease outcomes like type 2 diabetes, heart disease, hypertension, stroke, etc. But we have also shown that they are still at higher risk of some non-metabolic diseases, for example, osteoarthritis or deep vein thrombosis, etc. So I use all this data, and I have four main actually objectives. One is to find causal effects of obesity on risk of disease, and basically to see whether people, because data shows that different people are at different risk of, I mean, I'm talking about obese individuals, they're at different risk of developing disease outcomes. And by identifying subtypes, we can basically predict risk of developing disease in each group of individuals based on their genetic makeup. And also I use this genetic data to predict response to treatment, because some obese individuals, some people with obesity respond well to calorie restriction diets, some respond better to some tablets, and some of them need basically surgery, bariatric surgeries. So it's very important to basically predict which treatment works better for which group of people. And finally, by using this genetic data, we'll be able to identify more therapeutic targets. And this is important because the current approach is based on a one-size-fits-all model, and I think this research is needed to basically make us closer to a precision medicine in obesity. So if you could please move to the next slide. I use different techniques and methods, mainly based on genome-wide. I use data from genome-wide association studies, which is the cheapest way of identifying genetic factors for different diseases and traits. I also use whole genome and exome sequencing, and the data is available on 500,000 individuals from, again, the UK Biobank study. And this is really important because we can do lots of different things to identify genetic cause for common or very rare diseases, and also identify causal genes and hopefully therapeutic targets. I use, as I said, machine learning algorithm for clustering phenotypes and genetic variants. I use data from MRI scan data, mainly from abdominal scans, to understand the underlying mechanism for storing fat in a wrong place. For example, as I say, in the liver or pancreas, in the muscles. And I'm interested basically in all the methods that are used in precision medicine. In terms of a statistical method, I use a method which is called Mendelian Randomization, and this is the cheapest way. It's similar to randomized controlled trials, but we only need genetic data, and we can basically identify causal risk factors for any disease outcome if the data is available. And I use also genetic data to basically generate genetic risk models to predict risk of disease. I also use gene expression analysis, fat biopsy, and recruit by genotype studies. I've been successful applying in some grants from European Foundation for the study of diabetes, from Wellcome Trust, from British Heart Foundation, from MRC, and currently my main funder is Diabetes UK, which has funded me to basically identify mechanisms that protect obese individuals from developing risk of disease using genetics. Thank you very much.
Thanks very much. It's nice to talk to everyone about research. Don't usually do that, so it's great to have the opportunity. Bit of background about me. I'm actually first degree was chemistry, biochemistry, then PhD in engineering, and my research has always been at the interface of the sort of physical sciences, engineering, and biology. So I pretty much measure things. That's what I do. But a few years ago, I got involved with the non-invasive disease diagnosis using the analysis of volatile organic compounds. And this is pretty broad. There's an awful lot of things you can do with this. It's quite a new topic, probably 20 years or so. And I started this right at the beginning of that, really. So it's been quite an interesting space to work in. So just to be clear what volatile organic compounds are, anything with a vapor pressure. Now, they're typically really small molecules, less than about 400 Daltons, because anything bigger than that won't have a vapor pressure. And you can either detect them from breath or from the headspace of a sample, which can be liquid or solid or whatever. And just to be clear, the kind of samples we're talking about is things, various body fluids. Could be off skin though, so tissue. So it could be, you can actually measure directly stuff that comes off skin, phlegm, pus, blood, urine, feces, the whole lot. We can measure volatile organic compounds that come off that. And you can use that to either pick up a localized problem, issue, whatever, physiological thing that's going on around a particular organ, or a systemic thing where you've got some kind of metabolic disease or some systemic infection, so you can pick this up as well. So why on earth would we want to do that? Well, there's quite a lot of reasons that measuring volatile organic compounds can actually tell us quite a lot about the state of any biological system. There's quite nascent in this, so there's a lot that we don't know, but we're starting to learn a lot more about this. If we're using this in developing diagnostics, these tests can be made much less invasive than traditional tests. So rather than poking a big stick in someone and pouring out some tissue, you can just get someone to breathe into a bag, or you can get some urine and ask people to give you urine, and that will give you quite a lot of information about what's going on. The actual tests, the analytical technologies we use, I mean, I have an instrument in my lab down the corridor, which can analyze a sample in about a minute. So it's quite quick. And because of the speed and whatever, you can actually, I suppose it's how long is a piece of string, but you can make these tests if you know what you're looking for, and you know a specific compound or set of compounds that you're looking for. You can design sensors around them, which can be made very, very cheap and portable and easy to move around. So they're quite reasonable. And being non-invasive and what have you, usually patients are much happier than having a biopsy taken. And there's obviously the potential for point of care devices. So there's a lot of advantages. I'm not going to tell you the disadvantages today because this is a positive thing. Obviously there are some, otherwise everyone would be doing it. So we can take samples from all sorts of things. As I said, breath, urine, skin, etc. But there's lots and lots of applications, and I've had funding from pretty much all of these things in here. So I've done a lot of work with diabetes. It's quite interesting how volatile organic compounds change in diabetics, depending on whether they're hypo or hyperglycemic, and there's quite a nice correlation there. We heard a bit about tuberculosis and other infectious disease monitoring. So I've had funding from DEFRA to do detect TB in badgers and cattle non-invasively using this approach. And one day I might give a talk on when things go wrong. So I've got some great footage of trying to breathalyze a cow, which is quite good. Which will, and it was my former boss, and I had a good laugh at that one, I have to say. Anyway, then I've done a lot of work on inflammatory bowel disease. So the differential diagnosis between Crohn's disease and ulcerative colitis is pretty difficult clinically to do without sticking probes into people, and even then it's not straightforward. But the actual treatment for Crohn's and ulcerative colitis, although they present similarly, is different. So Crohn's, in many cases you can treat that just using diet. People might not like it, but you can treat it using diet. Whereas ulcerative colitis, diet's never really going to solve that problem. You're going to have to give some immunosuppressants and whatever. So actually being able to differentially distinguish between those two conditions in a non-invasive way is quite a good thing. Cancer, loads of different cancers have different volatile signatures. So we've done quite a bit of work on that. Food intolerance, identifying what food intolerance causes it, symptoms and whatever, and working out what it is that you're allergic to or intolerant to. Dental applications, that's an interesting one. Just done a project with an endodontist who noticed that when he was doing his root canal procedures, there was a strange odour in the room over the course of a day. Turns out they use bleach, and it causes all sorts of nasty reactions with the rotting stuff that goes on in the tooth, and it produces all sorts of nasty, very toxic things, one of which is formaldehyde at 50 parts per million. And this stuff is infusing the room, and you can just imagine what health and safety would say about that if they knew about this. So that's something that we've recently published. A lot of animal health monitoring, as I say, with DEFRA funded projects, and I've done a lot of work with horses as well. And then wound healing and infection. So just going to actually just signed a project to get some funding to look at wound healing and to see whether you can detect whether it's wounds infected just by looking at the volatile compounds that come off a dressing. So I think that's all I want to say. The only other thing is about the funding that you can get for this pretty big range over the space of quite a few years. I've had funding, as I say, from DEFRA, European Union, Juvenile Diabetes Research Foundation, EPSLC, BBSLC, Wellcome, the whole range. So this is quite broad and pretty much worked across a lot of different sectors with that. But anyway, that just gives you an idea about stuff I've done.
Okay, so that concludes our presentations for today. I should just say there are a couple of other members that unfortunately couldn't be here. We have Dr. Uday Kishore, who works on pathogens and understanding the mechanisms that promote pathogens in different diseases, particularly in the context of the lung, and also Dr. Raha Pazoki, that works on sort of large-scale identification of genetic makeup for certain diseases. So she has a particular interest in diabetes and other metabolic disorders, using large data sets for that. So unfortunately they couldn't join us, but I'm sure that they will present some of their research at a later date. So I don't know if there are any questions from anybody. I don't think I can see anything. Hanya, do you see anything in the chat?
No, there's nothing in the chat. No.
I don't know if anybody has any particular question in the room. If not, okay. We also are having a sort of open day on the 19th of May for those who care to join us. Some of our younger members of the centre are going to present some of their data in oral presentations as well as poster presentations. That will go up online with details for that. It just remains for me to say thank you very much to everybody that presented and to everyone for listening. Thanks very much.